Hi everyone. In this video, we are going to be talking about cellular respiration. Once again, I just stopped recording on the redox video and we're just going to get straight into it, having the time of our lives. So it really is fitting that the previous two videos I did, within the last couple hours were, is this good? Yeah, I might zoom in a little bit more. Sorry, got distracted. The last few videos we did were cell membranes and redox. That is the perfect lead into cellular respiration because cell membranes are going to come into play both actually at the beginning of glycolysis. We won't get into much detail so much about it. It's more like an application, but especially when we talk about the electron transport chain, we're going to really, really hone in on membrane mechanics and membrane properties. Okay, so for cellular respiration, once again, we need that sort of overview. So this is all about breaking down sugars. So break down sugar to harvest energy and material to do stuff. Okay. Keyword harvest. This is usually a place where students lose points on exams. The professors love to write questions that are all about how organisms create or make energy in cellular respiration. This is not true because that directly violates the laws of thermodynamics. Remember, energy can neither be created nor destroyed. It is converted. What organisms do is they convert the energy that's stored in things like sugars into energy stored in things like ATP. It is a conversion or a harvesting of energy. Organisms do not create or make energy. Okay, now we're going to focus on a few different molecules here. They're usually ATP and ADP, NAD+. That's not a good. There we go. NAD+, NADH, FAD, and FADH2, less so. And finally, coenzyme A /acetyl-CoA. All right, let's take a look at each one of these. So ATP stands for adenosine triphosphate. So we're going to write out our three phosphates because tri does in fact mean three. Did I get three? Yeah, I did. Okay, and then adenosine. It's a big old nitrogenous base. So the thing that should go through our mind is, three phosphate groups, a nitrogenous base, and a sugar in the middle. This is a nucleotide. Okay, this is ATP. All right, so ATP can be broken apart into ADP and an inorganic phosphate. So here's our inorganic phosphate. Here is our ADP. This is done through a hydrolysis reaction. For the sake of time, I'm going to draw an A there. That stands for adenosine. Okay. So if we add in water, we can break apart ATP into ADP and P sub I. What that also means is we can remove water and we will get the formation of ATP. Now, we need a way to describe this in terms of thermodynamics. And what I want you guys to think about when you see these phosphate groups is you should think, wow, oxygen is really, really greedy. It's sort of that toxic, greedy person you know in your life. In other words, phosphate groups are you and your toxic ex. So, let's consider this example. Remember, you are ADP. You're the bigger person. Your toxic ex is the piece of mind. They're the smaller person, right? You're the bigger person. Okay, we're all clear? Cool. If I want to get you and your toxic ex in the same room, do you think that's going to be relatively easy for me to do? In other words, do I think that's going to spontaneously occur? Probably not, right? Like, if I want to get you and your toxic ex in the same room, I'm going to need to put in a ton of energy, ton of effort to get y'all in the same room. So what that means is smacking together ADP and P sub I is going to require a lot of energy. In other words, the delta G is greater than 0. It is an endergonic reaction. Okay, let's say you guys are in the same room, right? I have you and your toxic ex in the same room, and I open that door and I allow y'all to leave. Chances are y'all are going to rush out of there. It's going to spontaneously happen where y'all go to separate directions and probably say some inappropriate things to each other as you're going your different ways. What that means is as we break apart ATP, this is going to release energy. The delta G is less than 0. This should be ingrained into your brains. Okay? Very, very important you have that memorized. Hydrolysis of ATP is exergonic. Formation of ATP is endergonic. Okay? All right, focusing on NADH. So NADH, it is actually built off of an ADP molecule, where that ADP, through its second phosphate group, is attached to another ribose sugar, and that ribose sugar, I need to give myself a little bit more room. There we go. That ribose sugar is then attached to a small little ring. Okay, so this here is our NAD+. And if we remember back to the redox, and I told you to memorize that NAD+ is the oxidized state and NADH is the reduced state, if I want to reduce NAD+ to NADH, I need to add in a reducing agent. Similarly, if I want to oxidize NADH to NAD+, I need to add in an oxidizing agent. Okay, I can then draw in my molecule. So ADP, ribose, there we go. And what we end up with is this guy, where one of these carbon-hydrogen bonds up here is the one over here, and then the second one is the one we added from our reducing agent. Now, similarly, we want to be able to describe the delta G of this. But as we already learned, the delta G is equal to negative NF delta E. We have, let's say, going from NAD+ to NADH, we have the reduction half reaction, but we do not have the oxidation half reaction. So, in order to determine the delta G of a reaction involving NAD+ and NADH, we need to know who the other player is. Is it a reducing agent? Is it an oxidizing agent? We need the full reaction in order to determine the delta E and therefore the delta G. Okay? So, from this information alone, we cannot determine a delta G. So, in other words, the delta G is going to determine based on the other half reaction. In terms of coenzyme A, it is a very, very large molecule. I don't have it memorized. It's massive. What is sort of the tall tale is you will see this thing sticking out the end. Fun fact, this is a derivative of the amino acid cysteine. Okay, here's its N terminus. The C terminus was originally here. It was removed through a decarboxylation reaction. Anyways, can I get rid of this? Cool. It's a derivative of cysteine. Now, what can happen here is if we have a 2-carbon group, especially due to that carbonyl, we can actually attach that. So if we add in an acetyl group, in other words, our 2-carbon acetyl group, we end up with, there's our acetyl, here's our 2 carbons that are being carried. And it will once again be attached to the rest of the coenzyme A molecule. So this here is acetyl-CoA. This here is coenzyme A. To go backwards, right, from acetyl-CoA to coenzyme A, we need to remove that 2C acetyl group. So what you need to know is coenzyme A is a common 2-carbon carrier. All right, these are all our players. We have a few more overview things to do. Okay, the first one is our overall reaction. Okay, the overall is C6H12O6 plus 6O2 goes to that is not a 6. 6CO2 plus 6H2O. Okay. And energy. As it's written now, this is a combustion reaction. We have a hydrocarbon and react it with oxygen. So in one step, this releases a lot of energy. Actually, we don't even need to say in one step. We can just say this releases a lot of energy. If this is in one step, if performed in one step, this is going to kill the cell. It's too much energy released at once. It's going to absolutely fry your cell. So the solution is a multi-step. Multi-step harvesting of energy. And if we were to look at the graphs of this, okay, so let's say we have 2 graphs. Remember, the reactants are the exact same. Glucose, oxygen. Our glucose, oxygen as our reactants. Carbon dioxide and water are our products. Remember, the delta G of reactants, the delta G is equal to the free energy of the products minus the free energy of the reactants. Whether or not we do this in one step or multiple steps, the overall delta G is still the same. Here's it in one step, or we could do it in sequential steps. Something like this. Once again, delta G is identical. all that's happening is instead of having a one massive release of free energy, it's stepwise, right? It's releasing that little bit, and then that little bit, and then that little bit, and then that little bit. And in the true cellular respiration, it's way more steps. This is just an abbreviated example. But the overall delta G is the same. Okay? All right, so now we can move on to our actual concept map. And like I've told you guys in discussion, this is sort of the thing that I love most about what you learn in discussion is these concept maps. They are immensely helpful and highly recommend being able to draw this out as much as you can. Spend time getting to learn your concept map. Okay, so this is going to start off with glycolysis. Okay, glycolysis is going to take in glucose and convert it to 2 pyruvate molecules. How this is going to happen is we're going to take 2 ATP and convert it to 2 ADP. We are also going to add in 2 inorganic phosphates and 2 NAD plus to give 2 NADH. And then finally, take 4 ADP and get out 4 ATP. This occurs in the cytoplasm. So for this class, for each of our processes, you need to know the ins, the outs, the where, and the why. Here's your ins, outs, and the where. And the why of glycolysis is to initiate sugar oxidation and really harvesting energy and electrons from that sugar. That's the goal of glycolysis. It provides you with a little bit of ATP, right? Its net ATP production is 2, right? 4 minus 2 is 2. Its gross ATP production is 4. So it gives us a little bit of ATP, and it also gives us a little bit of NADH that we can use later. Okay? Now, this pyruvate can undergo 2 pathways. First one is the reductive pathway. It undergoes reduction. Okay, it can also undergo oxidation. Now, why would pyruvate undergo reduction? Well, let's consider a thought experiment. If we just had glycolysis going, gradually what we would have happen is all our NAD plus is going to be converted to NADH. And if we run out of NAD plus, the reaction that requires it, which is glyceraldehyde 3-phosphate dehydrogenase, will stop functioning because it will lose the oxidizing agent. Okay? So we need to regenerate our NAD plus pools, and the first way we can do that is through fermentation, where we're going to take our NAD plus, not NAD plus, NADH, oxidize it back to NAD plus, and in the process, we are going to make lactate or lactic acid or ethanol. These are the 2 fermentative pathways we teach you. Lactate is 3 carbons. Ethanol is 2 carbons. So if you are talking in terms of carbon retention, lactic acid fermentation is actually more efficient. Ethanolic fermentation, you lose a carbon to carbon dioxide, and once you lose it, it's hard to get it back. Okay. All right. But like we said, this is kind of wasteful, right? We're just taking those electrons we just spent so long harvesting and then just getting rid of them. So maybe we can use that, right? Maybe we can find a way to put those electrons somewhere else, but still get more out of it. And so that's where we're going to go into the oxidative pathway. Oh, by the way, the reductive pathway is known as fermentation. And this is also in the cytoplasm. All right, in terms of the oxidative pathway, this is going to require transport into the mitochondria, assuming we are in a eukaryote. If we are in a prokaryote, this does not occur because prokaryotes lack membrane-bound organelles, including the mitochondria. All right, so what's going to happen is this pyruvate is going to get immediately oxidized. We're going to take our NAD+, and we're going to add in our friend, coenzyme A, and we're going to get out of it a CO2 molecule and NADH. Note that this is per one pyruvate. So one pyruvate, which is a 3-carbon compound. Glucose is 6 carbons. The one pyruvate enters what's called the pyruvate dehydrogenase complex. And that converts one NAD plus to NADH and produces one CO2. So if we have 3 carbons, we lose 1. We have 2 carbons, and hopefully alarm bells are going off. You see coenzyme A and 2 carbons left over. That should produce our good friend acetyl-CoA. Okay. I'll put this in parentheses. All right. So this is occurring inside the mitochondrial matrix. Note that mitochondria is often abbreviated MT. And now this acetyl-CoA can enter the Krebs cycle. During this, one molecule of acetyl-CoA takes 3 NAD plus and converts it to 3 NADH. It also takes, what's it called? Oi. So I want to do that next. Yeah, I will. Yeah. It will also produce one molecule of ATP. Now, depending on the very specific enzyme that does this, which if you're curious is the succinyl-CoA synthetase enzyme, depending on which one, it will either produce ATP or it might produce GTP. They are essentially analogs of each other, or they're essentially brothers. So you will often see it abbreviated like this. This is just saying that depending on which specific version of the enzyme you use, it will produce either ATP or GTP. They're chemically and biochemically very, very similar. Note that this does also require an input of an inorganic phosphate. Okay. And then finally, out of this, we get one molecule of FAD being converted to FADH2. And then out of this, we get 2 CO2. Okay, note that that is per one acetyl-CoA, and one acetyl-CoA is one pyruvate, and there's 2 pyruvate per glucose. All right, this is known as the Krebs cycle, or the TCA cycle, or the citric acid cycle, or the tricarboxylic acid cycle. There's like every single name possible. Call it one of them and move on with your life. This also occurs in the mitochondrial matrix. But if we, sort of consult ourselves back to our fermentation, this fixed our NAD plus problem. The oxidative pathway, although we extracted way more NADH, it actually made our NADH problem worse. So we have an issue. And the solution is a place where we can dump all these electrons, but get something out of it. And we are going to look to the electron transport chain for this. So the mitochondria has 2 membranes. It has its outer membrane and the inner membrane. We then have the space between the two membranes, otherwise known as the intermembrane space, and then we have the matrix. Remember, this is where the Krebs cycle is taking place. Okay. We then have a series of very large protein complexes. A smaller molecule called QH2 that is going to float sort of inside of the membrane. Again, another large protein complex. A smaller guy called cytochrome C, which is also a protein, and it will also sort of float around within the membrane. It is a mobile carrier, and these large protein complexes are immobile. This is complex 1, complex 2. Oh, that is not complex 2. That is complex 3. There's complex 3. This is complex 2. And here's complex 4. Okay. All right. So what's going to happen here is we're going to oxidize that NADH, and those electrons are going to be passed to complex 1. And I'm going to tell you right now that that redox reaction is exergonic. And what can we do with an exergonic redox reaction? Well, we can couple it to an endergonic reaction, which is exactly what we're going to do. So we are going to pump protons across. This is technically primary active transport because the energy is not coming from an electrochemical gradient. So sometimes you will also see primary active transport as it's coming directly from like a chemical energy source, versus secondary active transport is coming from a electrochemical gradient. So this is technically primary active transport. I forget in the cell membrane, I think I called it secondary. I don't remember, honestly, but it is technically primary active transport. The more important thing for us to know is it's proton pumping. So we are pumping protons across the membrane in an endergonic reaction. That is getting paid for by the oxidation of NADH by complex 1. And so we have coupled electron movement to proton translocation or proton pumping. And so what that also tells us is that the protons are going from a low concentration to a high concentration. That would result in an endergonic redox or an endergonic membrane transport. In other words, low concentration is a relatively high pH in the matrix, and in the IMS, we're talking about a relatively low pH, more protons. Okay, the electrons are then passed from complex 1 onto QH2. At complex 2, we take our FADH2 and we oxidize it to FAD. And I'll tell you now that this redox reaction is exergonic. In fact, all redox reactions in the electron transport chain are exergonic. This is what allows us to couple it to proton translocation. So both of these are going to reduce coenzyme Q to QH2. If you're curious, it's also called quinone when it's just Q, and quinol when it is reduced. If you're even so curious, this here is Q. And then if you add in 2 electrons and 2 protons, you end up with QH2, quinol. Okay, that's what that guy looks like. Okay, all right, so these electrons get passed from QH2 onto complex 3. Complex 3 then passes them to cytochrome C, and through those redox reactions, it pumps protons across. Cytochrome C then passes the electrons onto complex 4, which also pumps protons across, but we're going to hold off here. And let's go through once again a thought experiment. If complex 4 doesn't toss these electrons into anything else. Those electrons are going to sit on complex 4. And if complex 4 then stays in a reduced state, it can't then oxidize cytochrome C. Remember, it's not just one pass-through. It's continuously sending NADH and FADH2 through complex 1 and complex 2 to go through the electron transport chain. So if electrons pile up on complex 4, there's still more electrons coming. So what that means is cytochrome C is going to end up with electrons, and it can't do anything with them because complex 4 is going to stay reduced. And if cytochrome C stays reduced, it can't then oxidize complex 3. And if complex 3 can't be oxidized, it's going to stay reduced. If it stays reduced, it can't oxidize QH2. And if QH2 can't become oxidized, it can't oxidize complex 1 and complex 2, which means these two can't oxidize NADH and FADH2 respectively. And so then we're right back at our issue, right? We've pumped a little bit of protons across, but then this entire ETC comes to a grinding halt because we have no place to dump these electrons. In fermentation, we solved it by dumping them onto pyruvate. We no longer have that pyruvate, right? This here is a branch point. It can either go reduction or oxidation. It can't do both. And so what the electron transport chain is going to do, the ETC, it is going to dump these electrons onto a terminal electron acceptor. So the terminal electron acceptor in an oxidized state goes to a reduced state. Now, inside the mitochondria, we have a very specific terminal electron acceptor, and that is oxygen. So oxygen, along with 2H+, just for stoichiometry, is going to go to water. If oxygen is our terminal electron acceptor, this is aerobic respiration. If oxygen is not the terminal electron acceptor, literally anything besides oxygen, it is anaerobic respiration. Okay? So the ETC is going to directly produce H+, high concentration in the IMS and low concentration in the matrix. In other words, the ETC directly produces a proton motive force. And if we think about it, the exergonic flow of protons is going to be into the matrix from the IMS. So let's take advantage of that. And we're going to add in our last enzyme. This is ATP synthase. And what ATP synthase, I don't like that circle. ATP synthase is going to do is it's going to allow the protons to go through the membrane. Actually, technically it's like this, but whatever. So the protons are going to go through the sort of pseudo channel that exists here. And as the protons go down their concentration gradient and release free energy, ATP synthase is going to couple that to the formation of ATP from ADP and P sub I. So this protons going down its concentration gradient is known as oxidative. Not oxidative. It is known as chemiosmosis. What the? I don't know what's happening. Chemiosmosis. So that's the H+ ions going down their concentration gradient. And then the formation of ATP from ADP and P sub I using the electron transport chain is known as oxidative phosphorylation. Note that this stands in contrast to what's called substrate level phosphorylation. Substrate level phosphorylation is the synthesis of ATP from a phosphorylated substrate. So SLP occurs in the Krebs cycle and in glycolysis. Okay, to show you an example of that, I'm going to pick on one reaction in glycolysis. Let's do phosphoenolpyruvate. Actually, no. I'm going to do 1,3-bisphosphoglycerate. Okay. This here is 1,3-bisphosphoglycerate. And what's going to happen is we are going to make ADP, or we're going to take ADP and take one of these inorganic phosphate groups, this guy here, and slap it onto ADP to make ATP. And so we're going to be left with ATP and this molecule 3-phosphoglycerate. And the enzyme that does this is phosphoglycerate kinase. Okay. So here you can see we're making ATP where the phosphate group comes from a phosphorylated substrate. So the phosphate group and the energy required to make ATP comes from a phosphorylated substrate. In other words, this guy here. This is substrate level phosphorylation. There are 3 instances of it occurring, twice in glycolysis and once in the Krebs cycle. Okay. Once again, that is in contrast to oxidative phosphorylation, which is the production of ATP using the proton motor force created by the ETC. So to recap, once again, the ETC directly produces a proton motive force, which is then used by ATP synthase to make ATP. Note that the ETC does not directly produce ATP. It directly produces a proton motive force, which is used to make ATP. So indirectly, it's used to make ATP. But because of that sort of slight detachment, what that means is these can actually be decoupled from each other. What that means is if we were to provide an alternate pathway for these H+ to flow through, we can dissipate this gradient, the proton gradient, without actually generating ATP. There have been a few drugs that existed to do that. If you get bored one day, look up dinitrophenol. It worked so well that it had to be removed from the market because, well, people were dying. It worked so well at weight loss that it was killing people. It was sort of a crazy deal. If you get bored one day, look up dinitrophenol. Anyways, so yes, this is cellular respiration. Remember, you need to know the ins, the outs, the wheres, and the why. I was thinking about making a video about every single reaction in cellular respiration. It is currently 1:30 and in the morning, and I kind of want to go to bed because I have a lot of stuff to do tomorrow. So I might make it at another point in time. But yeah, if that comes out, of course, you guys will get an announcement from me on our Canvas page. The very last thing I'll say, just because I was looking at this and realized I never said it, oxidative phosphorylation is the main source of ATP for the cell. Glycolysis and Krebs cycle produce a little bit, but the majority of it comes from oxidative phosphorylation at the mitochondria. Okay, so that is where most of the ATP comes from. All right, as always, I hope you guys are, taking good care of yourselves. Make sure to eat healthy and get a lot of sleep. Don't do what I'm doing and staying up really, really late. It's bad for your brains. Eat healthy, get rest. Make sure to take breaks. Don't burn yourselves out, if you haven't already. And yeah, if you need anything from me, as always, you can shoot me an email, message me on Canvas, come talk to me in office hours. You can go to the other TA's office hours. There's review sessions. There's all these resources that, I hope you guys feel comfortable taking advantage of because they are there to help you. All right. I wish you guys the best with all your studying, in all your classes, in your research, in your extracurriculars. Maybe you're working at a homeless shelter, or maybe, at some sort of nonprofit or anything like that. Whatever's going on in your lives, I hope it's going well. And I'll see you guys soon.